Hello, everyone.
Hi.
Hi, welcome. We're just a couple minutes before 4 o'clock, or we're just coming on to 4 o'clock. So we'll give folks a couple more minutes to come in. You are welcome to turn off your video feed. I typically mute you as you come in. It just helps us stream a little smoother if your microphones are muted. If you want to speak, of course, you can unmute yourself. That's fine. We just try to keep the stream going as smoothly as possible. Well, my name is Dr. Piscopo, and I'm going to be your instructor for Public Health 240, which is microbiology for healthcare professionals. And because of the ongoing pandemic, we're going to be meeting for the course remotely. We're going to be meeting over Zoom. So welcome to the course. If you would do me a favor and type your first name into the chat for me. That's the way I take attendance at the beginning of our meetings. So just open up your chat function in your menu bar and then just type your first name in. And that will help me keep track each day that we're together of who's here and who isn't. Very good. All right. So our job today is to talk a little bit about the course and how the course is organized and what's expected of you in the course. And then we're actually going to start into our very first laboratory topic, which is just biosafety, sort of general safety procedures that we use really in a lot of lab spaces, but especially in a microbiology lab. So let's begin by talking about how this course is organized this semester. We're meeting for laboratory like this over Zoom. And of course, that means that you won't be able to get your hands into the materials yourself, but rather you'll be watching me doing certain procedures and techniques, and we'll be talking about those together. And we'll start a little bit later in the semester actually doing experiments. Again, with me doing the work of the experiment and sharing and with you as I go, essentially. And then we can talk about the data that we generate and the results we get, and so on. So our laboratory work is virtual and it's synchronous. That's the term we use to say that we meet during our regular lab time. So that's Tuesdays and Thursdays from 4 o'clock to 5:45. Now, the lecture part of the course is asynchronous. And of course, all that means is there's no specific scheduled time for lecture during the week. What I do is I post lecture topics for you each week in a form of a narrated video. And I personally post my lecture videos up to YouTube. I use YouTube because it's very reliable and there's no limit to how many videos I can post or how much space I can use. So I've been very pleased with them as a host channel, if you will. I do teach a lot of different courses. If you looked at my introduction information, if you've been in Canvas already, I teach at several different schools and I teach a lot of different courses. So I have lots and lots of lecture material posted to YouTube. So to keep things a little more organized, I've set up a playlist in our YouTube channel that's just for us. So there's a playlist that is titled PH 240, Microbiology for Health Professionals. And all of our video materials will be posted in that playlist on the YouTube channel. When you go there the first time, you might want to bookmark the playlist just so you can go immediately back there and quickly find the video material you need to find. Now, when I post videos, I don't post a lecture start to finish. I break it up into several smaller, I should say shorter videos. So, instead of posting a lecture that lasts an hour and a half or whatever, I post maybe 4 lectures that are 20 minutes each, but they're all the same lecture. It's just that I've cut it up into 4 pieces. So when you go onto the playlist, you'll see, for example, this week, the lecture topic is in 4 short videos, and they're labeled number 1 of 4, number 2 of 4, number 3 of 4, number 4 of 4. So you'll always know which videos you're supposed to be watching and how many there are in that lecture topic. I try to make it very obvious for you when you go in what the video is that you need to watch. So when you watch videos in here, you just want to act as though you're in the classroom. You're just going to want to pull up the video and sit and watch it and take notes, just like you were in the classroom. There is an added bonus in that you're allowed to use your lecture notes during the lecture quizzes in here and also during the lecture exams. So I want to encourage you to take as full and detailed a set of notes as you can from each lecture topic, because that way you'll have all the information you could possibly need when you sit down to take the exam. I want to take some of the pressure off you while you're taking the exams. So you're allowed to have your notes next to you while you're taking the exam. And hopefully that's incentive, not only to watch the lecture videos, but also to take a good set of notes for yourself while you're watching the videos. You'll also be doing something similar in laboratory when we're talking about a laboratory topic. You'll want to take a nice set of notes for yourself because you'll also be having what we call lab practical exams. It's essentially just an exam for laboratory material. So you'll have lecture examinations and you'll have lab examinations for the 2 different sets of material, basically. And you'll be able to have your lab notes with you when you take the lab exams. So again, it's absolutely worthwhile to take a nice detailed set of notes for yourself while you're either in laboratory like this or if you're watching a lecture video. Okay, so let's talk just a minute about how the course is organized on Canvas, because of course all of the course material that is not video material is up on Canvas for you. So anything that's just a written file is up on Canvas for you, and I've tried to organize it for you week to week. I want to make it really easy for you when you go to Canvas to find what you need to find. So you're going to notice that every week is treated like a module on Canvas. And inside each module for each week, in other words, I'm posting a to-do list for you. So you can open that up on, Sunday night or Monday morning, and there'll be a list of all the things you need to do that week for this course. Okay, that way you don't have to look around in different places to know what you're supposed to be doing that week. I know it can get really confusing very quickly when you're trying to keep your calendar straight, especially when you're taking online courses like this one. So Canvas is really sort of like your home base in here. You'll want to go to Canvas frequently. You'll want to, I would say, go to Canvas at least once a day, just to see if there's anything new that you need to see, like an announcement. And remember, you can change your settings in Canvas. If you want to get announcements, for example, sent to your cell phone, you can do that. You can change all kinds of settings for yourself in Canvas to make it as efficient for you as possible. So I'm going to pull up a screen right now to share with you. I'm going to pull up our homepage in Canvas. Just bear with me a minute. So on your screen, you should see our homepage for our Canvas course. It says microbio for health professionals. Health professionals is shortened. A few letters are missing there. So when you go into this course on Canvas and you click on home in the left-hand menu, you're brought to this page. So again, think of this sort of like home base for the course. And I say that because there's all kinds of information. There's information again about what's synchronous and what's asynchronous and things like that. And then there's some really important documents and links for you. You can see there's a link for our syllabus and schedule. There's a link for the YouTube channel where you're going to find our video materials, and then our course playlist specifically on that YouTube channel. Then you could see our information regarding Zoom meetings and so on. And then there's some information down the bottom about how you just get started in the course. Now, some of you have already gotten started, which is great. I'm pleased to see that. But if you haven't gotten started yet, what you're going to want to do is come in here into the course to the home page, and then you're going to want to click on where it says modules on the side menu. And that brings you to this page. So remember, the way the course will be organized is week by week. So you can see the first module is called Start Here. And then we see week 1, the week of February 15th. So that's this week. And then I've also got the second module completed for you, week 2, the week of February 22nd. I try to keep information on Canvas so that we're about a week ahead on Canvas than where we are in actuality. Now, there's a reason I do that. You really only need this information this week, but I post next week's information too, just in case you need to go a little ahead of schedule. Some students, because of their work schedules or their responsibilities at home, some students like to get a head start on things. So I try to keep us about a week ahead in materials, okay? Now, I actually have also just written down the third module, but there's nothing in it right now. So how do I get started? Well, it's just a step-by-step process to get started. You're going to want to click on this. This is will bring you to the syllabus and schedule. This is the link that helps you find your textbook. We are using an open source textbook in this course, which is great. That just means you don't have to buy it. You're free to use it online without any cost to you. It's a nice textbook. It's through this group called OpenStax. They produce lots and lots of online textbooks for students to use, including a microbiology text. Now, there is a note here because I have had some students want to purchase a hard copy of this book. It's about $50, at least it was the last time I checked. But it is possible to buy this book, to go to that website, OpenStax, and you can purchase the book there. Some people just like having a hard copy of a book, and I can appreciate that. I'm one of those people too. So if you'd like to buy the book, you may. It's certainly a nice reference book if you feel it might be helpful to you in the course and beyond. It's not a bad price for a reference book. But if you just want to use it for the course and you don't want to pay for anything, that's fine too. No problem there. Then step 3 is going to ask you to post onto one of the discussion boards, and it's called Introductions. And that board is set up just so we can introduce ourselves to each other. You know, one of the hard parts about being online like this is we don't get to be together in the classroom, so we don't really get to just speak casually to each other and get to know each other. So this is an effort to sort of work around that. Um, it's a place where you can just tell your classmates a little bit about yourself and, you know, post a picture of yourself if you'd like, and talk about things like, you know, what you're studying and what you hope to do when you're finished with your degree. And, are you in a relationship? Do you have kids? You know, things like that that you might want to share with the group. Step 4 is going to ask you to complete a short survey and then submit it to me. The difference between these two things is the introductions board is sort of a public place for the whole class. So whatever you put on the introductions board, everybody can read and reply to and so on. But I've also put together a survey that's for my eyes only. And that's, again, I want to get to know you a little better and I want to get to know the class a little better. So I've put together a short list of questions, you know, again, about who you are as a student and what you hope to do in your life and things like that. But you can rest assured that I'm the only one who looks at the student survey if you want to tell me anything that's more of a private nature. When you finish with your survey, and of course you'll have to download the document, which is posted right here. You'll have to download this to your computer, and then you'll have to save it, and then you can upload it to Canvas, and you'll get a few points for that. You'll get 5 points if you do that. And this is the link that you use right here to submit your survey to me. And I would just ask that you try to do that survey sometime this week. If you could get that to me by Sunday, that would be great. So looking at this week for us, I'm going to go ahead and click on our to-do list. Again, there's going to be a new to-do list each week. And it's just a place where I've written down everything you need to be doing that week. So you can see for this week, you have one lecture topic. And that topic is called Introduction to the Microbes. And again, any video material, and these lectures are going to be in video format, is posted on YouTube in our playlist. This week, since it's our first week, there's only one lecture topic. But during most weeks, there will be 2. And again, each one of those lecture topics will be broken into short videos. So you don't have to sit and watch an hour and a half long video. Take a detailed set of notes while you watch each lecture. And then there's a short lecture quiz on Canvas associated with each lecture. All right, so this week you'll be taking your first lecture quiz. These are not, high stakes quizzes or anything. These are little quizzes. They're worth 5 points. There's typically 5 questions, and it's all related to the lecture that you just watched. And again, you can use your lecture notes that you've taken while you take the quiz. So it's a nice way to check that information is in your notes. Typically, when you have 2 lectures during the week, one of your quizzes will be due Wednesday night, one will be due Friday night. And this week, since we only have the 1 lecture, the quiz is due Friday night. And I typically set all of our assignments to be due at midnight. It's just easy. So you can see down at the bottom, it says we have 2 Zoom meetings this week on Tuesday and Thursday. Some weeks this semester, we will only meet once during the week. So you have to, again, check in your to-do list each week so you can see whether we're meeting on Tuesday or on Thursday or on both days. And then for each of the laboratory weeks that we do, there'll be some homework. So you'll have one homework assignment each week based on what we did in lab. And those are due by Sunday at midnight. Now, there's a little bit of a difference between the lecture quizzes and the homeworks. They're both designed to make sure that your notes are in good shape and that you've understood the things that you've been watching and reading and so on. But the lecture quiz is a real quiz. The lecture quiz, when you sit down to take it, it's timed. You have 30 minutes to answer these 5 questions, and then you have to submit the quiz. Again, 5 points. The laboratory homework is also worth 5 points. Typically, there are some exceptions. But the lab homework, you can go in and out of as much as you want during the week. So the lab homework is not set up like a real quiz, even though Canvas calls it a quiz. The lab homework is more like, a homework. So you can go in and maybe do 1 or 2 questions, and then you can save that work and go do something else, come back the next day, and maybe do another couple questions, however you want to do it according to your schedule. So each week you'll have those 2 types of assignments to do. You'll have lecture quizzes to do, and you'll have lab homework to do. The lecture quizzes are actual quizzes. So you want to, when you sit down to take it, you have to finish it. You get 30 minutes, which should be more than enough time. But you have to just sit down, do it, submit it. But the lab homework's different. The lab homework you can do at your leisure. You can do some of it and then come back later and do a little more of it and so on. Does anybody have any questions about anything I've said so far? Like I said, I know several of you have already gone into Canvas. You've already gone through your Start Here module, which is great. If you haven't, just, you'll want to get in there, as soon as you're able today or maybe tomorrow and get started on things. We don't have a whole lot of work this week just because it's the first week. So I try not to overload you on the first week of class. All right.
Sorry,
Yeah,
I have
go
a quick
ahead.
question.
Yep.
So are we going to be like actively using the OpenStax like our textbook online, or is it more of like just like watching the lecture videos and stuff like that?
That's a great question. So the way I use the textbook in this course is as a supplement. And what I mean by that is all of the material that you need to know in this course is in the lecture videos and our lab periods together. So that means that there's not going to be anything on any quiz or exam that we don't talk about, either in the lecture videos or in our lab periods. Now, the textbook is a great, like I said, it's a great reference. It's a great textbook. And I encourage you to read the associated chapters as a supplement. In other words, maybe you watch a lecture video and there's a particular point that's not real clear to you. You can certainly go to the textbook and read what they have to say about that subject, and you might get a little clarification from that. Maybe you want to read a little bit more about something that I talk about in lecture, and you can go to the textbook. But there's a lot of information in the textbook that we will not be talking about in this course. So it's important to remember that anything that you read in the textbook that we don't talk about, you're not responsible for. Okay. The textbook is very thorough, and it's written not only for undergraduate students like yourselves, but also in some sections, for people who are in graduate school. So there's more information in the textbook than what you're going to be held responsible for. You do not have to read every chapter in the textbook. All right. I personally find that puts too much work on you. I would much rather that you spend the bulk of your time getting watching the lecture videos, getting a really good set of detailed notes made for yourself, and making sure you understand everything that's in the lectures. That's really the key thing. And of course, for the laboratories as well. I would rather you spend time doing that than forcing yourself to read every chapter that's listed in the syllabus. So think of it as a supplement. Think of it like frosting on the cake. If you find it helpful, by all means, go to the textbook and do a little reading, but don't feel compelled to read every chapter that is associated with the lectures. Okay. Good
Okay, thank
question.
you so much.
Yeah, good question. All right. I'm just going to jump back into Blackboard here for a minute. I want to point out a couple other things. Again, whoops, hold on just a sec. I've pulled myself out. I want to point out a couple of the things that you're going to find in our course on Canvas in the modules section again. So if I go down to the module for this week, here's our to-do list again. You can see I've posted what's called lecture slides only for our lecture topic, which is, again, Introduction to the Microbes. Some students like to have a copy of the PowerPoint slides that I go over in the lecture videos. And that's what these are. So in other words, this is the slides that I use in the lecture video that's posted to YouTube. It's just the slides. There's no narration. This is not a video. This is a PowerPoint file. If you're the kind of student that likes to use those slides while you take notes, some students like to open up the slides and take their notes right on the slides while they watch the lecture videos. If that's helpful to you, that's why those are there. So there's nothing you have to do with the slides. They're just here if you find them helpful. If you want to, for example, go back and look at a slide that was talked about in a lecture video, you can do that as well. Here's the lecture quiz that you'll want to take by Friday. And then you'll notice the laboratory material. Here are the slides that we're going to be using in lab this week. So again, it says lab slides only. In other words, these are just the slides. This is not the video. All of our video material is on YouTube. Anything that's in the form of a video is posted in our playlist on YouTube. And that's just because there are limitations to how much we can store on Canvas. And so I only keep, regular files on Canvas, and all of our video files are on YouTube. Here's the first set of laboratory questions I mentioned. Again, 5 points, and those will be due by Sunday at midnight. So the other screen I'm going to share with you right now is I'm going to pull up our syllabus. Again, just give me a sec. It takes me just a sec to switch over between the screens. So the link for this syllabus, which you should be looking at now on your screen, I've actually scrolled down to the schedule in the syllabus. So most of the course information is all above this, if you look at the syllabus. And then down at the bottom, you get to the schedule. This is a tentative schedule. Sometimes we get off schedule a little bit. You know, we get a little behind or we get a little ahead. So everything's always tentative. But this is how the semester is laid out if everything goes smoothly. Now, I want to show you how you read this. Again, you can find out in the to-do list everything that you need to do that week, any one week. But if you want to look ahead, pull up the syllabus and you'll find the whole schedule for the semester listed. Okay. So over here, again, it's listed by weeks. So here's the first week. It'll tell you what lecture topic is scheduled. And again, in most weeks, there are going to be 2 lecture topics. This week, we only have 1, Introduction to the Microbes. It'll tell you what chapter in the textbook is associated with this lecture. Again, if you find that helpful and you want to go into the textbook and do a little reading, but that's not a requirement. And then it will tell you what's going to happen in our Zoom meetings that week. So you can see on Tuesday, introduction to the course, and then Thursday, we're going to do some of this biosafety type material. In fact, we'll probably start this biosafety material today. I think we'll have enough time. This last column can be a little bit confusing. I told you a minute ago that every week you're going to have lecture quizzes and you're going to have lab homework, 5 points for each one of those. Sometimes you have additional work to do, and that's what I've got in this last column. So for example, in addition to the lecture quiz and the lab homework this week, there's that student survey that you have to complete. Okay. Again, this is all in the to-do list for this week. But if you want to look ahead, you can pull up this schedule in the syllabus. For example, you might want to write down when the lecture exams and the lab exams are happening. You might want to record that in your own personal calendar so you know when those are coming up. You may want to write down when we have lab and when we don't have lab in your own calendar. And I can appreciate that. So feel free, to use this for that purpose too. For example, next week, week 2, we're going to meet for lab on Tuesday, but we're not going to meet for lab on Thursday. So you can put that into your own schedule as you need to. So the syllabus is right up on that home base page in Canvas for you. And it's also, if you go into the Start Here module, there's also a link that takes you to the syllabus, just so you won't have any trouble finding it. The other thing I just wanted us to take a quick look at is our playlist on YouTube, in case you haven't been there yet. I'm going to share my screen with you again. Let's see if I can bring this up. Bear with me for just a minute. All right, share please. Okay, good. So at the top of your screen, or on your screen, I should say, you should see this. Up in the left-hand corner, the upper left-hand corner, you should see the YouTube symbol. I have YouTube Premium, which is why it says that. I don't like watching ads when I'm on YouTube, so I have YouTube Premium. I'm at our playlist right now. All right, so when you click on our playlist, and you can see the title of it right here, PH 240, Micro for Health Professionals. This is where all of our videos will be. All right. Again, you can go to the channel. That's not a problem. I'll show you what the channel looks like. It's called Science Science. It has this bright pink banner, so you can find it easily. And if you click where it says playlists, you can find our playlist, but you're also going to find a lot of other playlists. So if you go to this main page for the channel, you can find our videos, but they're going to be in and among lots and lots and lots of other videos, okay? Because I teach lots and lots of different courses. So if you come to the Science link, which again is on our home page, and then click on playlists, you can see I have lots of playlists for different courses. You're going to want to click on our playlist, which is this one right here, PH 240. Now you can see that our lecture topic for this week is broken into 4 shorter videos. Each one of these is between 20 minutes and the last one is 35 minutes long. So it's all one lecture. I've just split it up into 4 shorter videos. Now, the other thing you're going to notice is I also post our Zoom lab meetings up here because they are recorded. Obviously, today's our first meeting, but I've got one lab video up here that we're going to be using this week. I will label any videos that are related to lab with lab right at the beginning, so you know that they're lab related videos. The important thing to remember is when you go into this playlist, there's going to be more videos here than you're responsible for in this week. Remember, I try to post next week's material too. So just because a lecture is posted to the playlist, it doesn't mean you need to watch it immediately. Go to your to-do list and see which videos you're supposed to be watching this week. Okay. And if you'd like to get ahead and you'd like to watch something that's due next week because your work schedule is crazy or you know something else is coming up next week that's going to keep you busy, you'll be able to do that. But one of the nice things about Zoom is that when we meet over Zoom, Zoom records the meeting. And after we sign off today, within an hour, Zoom will process that recording and will send it to me. And then I will post it into our playlist. And the reason I do that is because sometimes, just like with lectures, sometimes when you're watching a lab or watching me doing an experiment or whatever it is we're doing over Zoom, sometimes when you're taking notes, you might miss something. So if you need to go back, you can pull up that video and you can review whatever you want to review from the meeting. And I also recognize that from time to time, students have to miss one of our Zoom meetings. I would ask that if you need to miss a Zoom meeting, please let me know. Send me a quick email and let me know as soon as you can. If you're not feeling well or you've had something come up so you can't meet with for Tuesday and Thursday, send me a message and let me know. And then you can just pull up the recording and watch it so you don't miss out on the information we covered in lab. Okay. Again, I try to make it so that you have everything you're going to need to be successful in the class. Okay. The trick to being successful in here is keeping up with the work. If that means setting aside a scheduled block of time, like where you're going to watch lectures or where you're going to take your quizzes, do that. You'll do yourself that favor. Because as I'm sure you know from your online experiences during the pandemic, it's very hard to stay motivated when you're taking a course online. When we're in person, you just have to get yourself to the classroom and then the lecture happens. But when you're taking a course online, you have to motivate yourself to sit down and watch the video. So do yourself a favor as much as possible and try to organize your weeks so you know exactly when you're going to be watching lectures and when you're going to be doing your lab homework and so on and so on. I know everybody's really busy. Everybody has a busy schedule. Everybody's working. It's very hard, and it's especially hard right now because we're all very stressed from this pandemic. And that's it's a good time for me to mention that I want you to know that I recognize how stressful this is. I know that you all have been through a terrible year and you've had your lives upended. And I know that it was not your intention to take these courses remotely. Um, I completely recognize that. And I want you to know that I'm here to help you be successful in the course. So I need you to keep in touch with me and let me know how you're doing. And if you start to get overwhelmed, if you start to get behind, let me know. If you can't get a lecture quiz done because you have 50 other things you have to do, send me a message. I will give you an extension. Okay. But you have to talk to me so I know. You have to let me know that you're feeling a little overwhelmed. And by all means, if heaven forbid you get ill or somebody in your family gets ill and your life gets disrupted, please let me know. And we'll work to get you, to help you get back on schedule. Okay. But I take that very seriously. I take my job very seriously. I'm your instructor and I look at my job as being here to help you be successful in the class. Okay. And this is a good point to mention. The best way to send me a message is through Canvas. In other words, if you go into Canvas, into our course, you can click on in the menu where it says inbox. If you've never done this, it's just a place where you can send email to people in your class. Um, the reason I say to do it through Canvas and not just through your regular email is because when you send me a message through Canvas, it goes right to the top of my inbox. As you can imagine, I get inundated with emails. I know you get inundated with emails too. I'm teaching at 3 different schools right now, so um, my emails are just overwhelming at times. I want to get your message though. I want your message to come up to the top. So that will happen if you send it to me through Canvas. And I don't have to wade through all the other emails that don't matter in order to find your message. Okay. So again, if you've never used the inbox function in Canvas, um, you'll want to take a look at that because that's how you can email people in this course, including me, directly from Canvas. It's actually a nice little feature of Canvas. All right. Let's see what else I wanted to mention. Um, you do have a project that we'll be doing this semester. It's not a big project. Please don't worry about it. But it is an opportunity for you to do something outside of an exam that will allow you to earn a nice big chunk of points. It's called the pathogen project in your syllabus and schedule. And it's basically an opportunity for you to do a little reading about one particular type of pathogen. And if you're not familiar with that word, a pathogen is simply an organism that's capable of causing disease. So there are, of course, many bacteria, many viruses and fungi and other types of microbes that we'll be talking about this semester. There are many pathogenic microbes, and the pathogen project basically allows you to choose from a list um, which pathogen you'd like to investigate, and you're basically assigned a set of questions that you need to answer about that pathogen. Um, Again, don't be too worried about it. It's not a heavy project. It's not going to take hours and hours and hours of your time. If you're interested in knowing when it begins and when it's due, you can find that on our schedule. And we'll be talking more about it as it gets closer. Something else to remember, this is always important. If we're ever in a meeting on Zoom and the screen just freezes, or if you get dropped or something like that, um, gosh, if you're ever taking an exam, again, over the internet, because we're remote, and you get dropped and you lose your exam, don't panic. All right. Don't ever panic in this course. We will figure it out. All right. That's the take home message today. We will figure it out. From time to time, it does happen. Students will be taking an exam in Canvas and their Wi Fi will go out. Or I've had occasions where Canvas has kicked students out, halfway through the exam and then won't let them back in. Don't worry, don't panic. send me a message, tell me what happened, and we'll get you back into the exam. Um, and if you get kicked out of a Zoom meeting like this one, remember you can always pull the recording up of the meeting later on. So even if you miss a lab, it's not a big deal. You can get the recording of the lab later and you can figure out what you missed. I want this course to be one of your least stressful courses this semester. Okay. I want to help you be successful. And I do, I genuinely mean that. Okay. I think that's all I needed to mention today. I'll open up, see if anybody has any questions. If you weren't here at the very start of our meeting and you haven't yet, go ahead and type your first name into the chat. That's the way I take attendance during our meetings. So I'll do that every time we meet. I'll ask you to type your name into chat right in the beginning. And then anytime I ask you questions during our meetings, you can either answer in chat, you can type your answers out in chat, or if you'd rather, you can unmute yourself and answer the question. Either one of those is fine. Sometimes I will call on people. I don't do that very often, but sometimes I will call on you to answer something during lab. So whatever makes you most comfortable. I know some people just aren't very comfortable talking out, speaking out on Zoom, and that's fine. That's not a problem. All right. If there are no questions right now and some come up to you, some come into your head later on, you can just message me and ask, and we'll I'll try to get them answered for you. What I'm going to do now, we're just about 4:45. What I'm going to do now is open up the set of slides that we're using for this biosafety topic. And we'll start working through that topic. So you'll want to go ahead and get your notes out, your notebook, if you don't have it out already. And we can get started on this. So again, at the start of this course, we do spend some lab time just talking about biosafety. This is a really good topic to master if you have any plans at all about being in or around a laboratory in your professional life. So that means, you know, if you're thinking I might like to work as a technician in a research lab, or I might like to work in a hospital, or I might like to work in a doctor's office, or any other health related type of a business, you're going to encounter laboratory spaces. So it's a really good idea to learn about the best ways to keep yourself safe when you're in a laboratory space. Now, some of you have already worked in labs. I know some of you have worked in research labs here on campus. Some of you are right now working in places that are healthcare related, hospitals and such. So you've already encountered, you know, diagnostic labs, or perhaps you've dropped off samples into laboratories. Maybe you're in a doctor's office and there's a lab in the office somewhere that handles certain samples. It's a really good exercise for us to review all of this sort of rules, written and unwritten, that help keep us safe in the laboratory. So I'm going to pull up our slides for biosafety in the laboratory. And remember, these are the slides themselves are available to you on Canvas if you'd like to take a look at them. I'm just trying to make this a little bit bigger for us. All right. So what you're looking at on this slide is some of what I would call just general rules about working in a laboratory. So these rules would apply to a microbiology laboratory, certainly, but they would also apply to really any other working laboratory. And the first thing to know is that any working laboratory is going to have a sign on the door that tells us what some of the hazards are inside that lab. This is what the signage contains. In addition to written information, it will also have one of these colored diamonds on it. So the next time you're on campus or the next time you're in the hospital and you're walking past a laboratory space, look on the door or next to the door for this diamond shaped signage with the 4 colors, because it has hazard information on it that you should be aware of. So in each one of these squares, there's a different hazard being communicated. And the way we use them is we put a number in the box between 0 and 4, with 0 being the lowest level of hazard and 4 being the highest level. So the red box, you might guess, is communicating any fire hazards in the room. You can imagine in some laboratories, like chemistry laboratories, there are a lot of chemicals in there that are highly flammable. So the fire hazard will be communicated here. And I know it's impossible for you to read it, but this slide actually lists what the 0 to 4 means. You can see that it talks about flash point. In other words, when will something flash and catch fire? Don't worry about the details of the 0 to 4 ratings. Just know that 0 is the lowest level of hazard, and 4 is the highest level of hazard. So if I walk past a lab and there's a 4 in this red box, I need to understand that there are things in that lab that are very, very flammable. There's a high fire hazard in that room. The yellow box is actually about about a chemical's stability. In other words, are there chemicals in there that are particularly unstable and can react under certain conditions? This white box doesn't always have a number in it. This is what we call special hazards. So if there's anything very unusual going on in the lab that creates a special hazard, that'll be listed here. For example, again, looking at chemistry, there are certain compounds that will ignite if they're exposed to water, things like that, special hazards. The blue box is about health. So if there are things in the lab that pose a health hazard to you, toxic things, in other words, that information will be communicated in the blue box. Again, with 0 being no hazard and 4 being high, the highest hazard. So this is an international system. You will find these diamonds on laboratory doors, not only in our country, but all around the world. It's an internationally recognized system of communicating hazardous information. So you know, if you work in that lab or if you're coming into that lab to drop something off, exactly what your risks are in that room. So that's the door signage that you need to be familiar with. The second thing I've got listed, I have in capital letters. It's super important. No food or drink in laboratory spaces. This again applies to all laboratories, particularly a microbiology laboratory where we're handling living microbes. It's just not safe to eat and drink in a laboratory because we're handling chemicals and cells and toxic things, and the risk is too high that we might accidentally consume something that could harm us. So it's just a general rule. If you want to eat or drink, you step out. You step out of the lab and eat in a different space. So similarly, you wouldn't want to put your makeup on. You wouldn't want to put lipstick on or anything like that around your eyes, because again, you could accidentally be applying some kind of a toxic chemical or some other dangerous substance to your mouth or to your eyes where it could really harm you. So we try to avoid doing any kind of touching around our mouth or our nose or our eyes while we're in a laboratory. again, just to keep ourselves safe. It's really important that you use gloves correctly in the laboratory. We're going to be talking more about this later on, but you have to know when to wear gloves while you're working. You also have to know how to put them on and take them off correctly. Gloves are really the one piece of PPE that you're going to find universally used in laboratories. Here's another really important one. We have to know how to dispose of materials that we're finished with. Again, in a microbiology laboratory, we're handling living microbes. And depending on the lab, we may be handling dangerous living microbes. So we have to know what to do with our waste materials. There are lots of things in a laboratory that just go right into the trash can or right down the drain. But there are lots of other things that cannot be disposed of like that. We handle lots of hazardous substances, so we have to have hazardous waste containers. We also, in the micro lab, have to be able to kill microbes once we're finished working with them. And we do that using a machine called an autoclave. We're going to talk at great detail about autoclaves later in the course. But for now, you should know that an autoclave is a machine that uses heat, high heat, and pressure to kill microbes and other cells. So it sterilizes anything that you put into it. When we're finished with microbial cultures, we autoclave them so we kill all the microbes and then we can dispose of them. Here's the last sort of universal thing for laboratories. Always wash your hands. Wash your hands when you arrive, when you begin working in a lab, and always wash your hands before you leave the lab. It's just good technique. It's a standard thing that we try to do in the lab. When you arrive to work in the lab and you're coming from home or you're coming from someplace else, wash your hands so you're not accidentally bringing something into the lab on your hands that could cause problems with your work. When you're ready to go at the end of the day, wash your hands so you don't accidentally bring anything home with you on your hands. It's just good practice. There's some other general things here, but we're starting to talk more about what we do specifically in a microbiology lab on this slide. Obviously, in any laboratory, you're going to have a workspace where you're going to be doing your work. And it's really important that you don't have a cluttered workspace. It's really important that you don't have all your personal items in your workspace. So I mean things like your coat, your backpack, your cell phone, your laptop, anything that's a personal item that you're carrying around with you. You're not going to want to have in your workspace. There are separate places for those personal items in a laboratory, and it's critical that you keep those things there. Remember, in a micro laboratory, we're handling microbes. You don't want to get microbes on your backpack. You don't want to get microbes on your laptop computer because then you could accidentally take them home with you and maybe cause problems. In the micro lab specifically, we always start the day by disinfecting our workspace. Always, always, always. You disinfect the desktop or the workbench where you're going to work. There are two disinfectants that are used in microbiology laboratories. The first one is alcohol, and the second one is bleach. Now, you'll notice that we don't use these full strength. Alcohol we use at 60 to 90% solution, so it's diluted. And the bleach we use at a 10% solution, so it's very diluted. Now, this is just plain old chlorine bleach. It's the same bleach that you would use in your laundry. Both of these solutions are sprayed onto the work surface and then wiped off with paper towel. and they will both do a nice job disinfecting. If there's any microbes present in your work area, these two solutions will kill those. Now, different labs use one or the other of these, sometimes both. It depends on the lab. I personally prefer alcohol to bleach only because somehow I always manage to get the bleach on my clothing. And as you know, bleach will take color out of your clothing. So I personally prefer alcohol. Any type of alcohol will work. So ethanol, propanol, isopropanol, any type of alcohol will work as long as it's diluted between 60 and 90%. Most laboratories use 70% alcohol, 70%. And it's confusing to people when you tell them that, well, you need to dilute the alcohol if you want to use it as a disinfectant. It doesn't make sense. It seems like 100% alcohol would work better, but it actually doesn't. The water actually helps the alcohol kill microbes. Um, it's been so interesting during this pandemic to watch the general public learn things about microbes, learn things about viruses. I don't know if you remember, but at least at the start of the pandemic, people had all kinds of questions about how to disinfect surfaces, right? I remember there was a real run on Clorox wipes for a while. I don't know if you can even get them yet. I don't know if they're even around yet, but it was impossible to find those little disposable wipes with Clorox bleach on them. Um, yeah, people have had sort of been forced to learn a little bit about how we disinfect during this pandemic, and whether they should disinfect packages, and how often they should disinfect and things like that. All right. So another really important thing, and this would apply to all lab spaces, but especially when you're working with microbes, make sure you gather all your supplies before you begin your work. If you're setting out to, let's say, you need to transfer some bacteria from one culture into a fresh culture tube. And when I use that word culture, I'm talking about some kind of a fluid or a solid material that has all of the things in it that the bacteria need to grow. It's all the things that they need to survive and to divide and make more bacteria. And those are what we call cultures. So if you're working on culture and maybe you're going to move some microbe cells from one culture to another, it's really important you gather everything that you're going to need before you begin working. The last thing that you want to have happen is have yourself, your hands all into some transfer procedure when you realize that you need a piece of equipment that you don't have with you. It's very disturbing to your work. It can ruin an experiment. So we always make sure we have all of our supplies together before we begin. And here's one of my favorites: label containers. Now, I'm talking specifically about a micro lab here, but this also can apply to all containers in all laboratories. Everything should have a label on it. Anybody should be able to walk into a lab, pick up a container, and know exactly what's in it. Again, it's how we stay safe. But in a micro lab, especially, some of our containers contain microbes. We grow microbes in the lab. So we use test tubes and we use petri dishes in order to grow these cultures of cells. And it's really critically important that those containers be labeled. So I've written down the information that you put onto these containers, obviously your name or your initials. Whoever made the container needs to put at least their initials on it. The date that your culture was made, the name of the organism that's growing in that culture, very important, And finally, what are they growing in or what are they growing on? One of the things we're going to talk about this semester is all the different kinds of media that we use in the micro lab to grow bacteria. And it's important that anybody in the lab who might come upon that container can pick it up and know exactly who made it, when they made it, what's growing and what it's growing on. Labeling is one of my big things in the lab. I always try to make sure that students keep up with their labeling. On this slide, we just have a list of some of the things that we might think of as good lab hygiene. I've already said that you want to keep all your personal things out of your workspace. I think that's probably obvious, but you also need to keep yourself presented properly in a laboratory. And what I mean by that is you want to wear appropriate clothing. Um, Depending on the lab where you might work, that may mean that you have to wear a lab coat like this person is in this image, but not necessarily. In some laboratories, the people who work there, the technical staff, wear scrubs. In some laboratories, they wear their street clothes. It just depends on the laboratory, and it depends a lot on what's being done in that laboratory. But regardless, even if you're only wearing your street clothes when you're working in the lab, you want to wear the appropriate clothing. You want to be careful that you're not wearing anything that is, let's say, particularly loose and flowing. You wouldn't want, for example, to wear a big bell sleeve on a blouse or something like that, because your sleeve might, accidentally brush your workspace and contaminate it. Similarly, you don't want to wear any long hanging jewelry. You wouldn't want to wear a long necklace because if you leaned over, your necklace might contaminate your work. Of course, you need to wear appropriate shoes. In any laboratory, that means your toes need to be covered. You want a closed toed shoe. So no flip flops, no sandals. Um, it's important because it's a safety. It's a safety feature in the lab. If you're like me and you have long hair, you need to keep your hair back and away from your face while you're working. So you have to tie it back in some kind of a ponytail or a bun. That's really important. Not only can your hair contaminate your work can contaminate your hair, and you can carry microbes home with you in your hair if it's, hanging and touching your work surface. The other big hazard with hair is fire. I can't tell you how many stories there are. Every year, there are stories out of laboratories around the world of people who accidentally set their hair on fire in the lab. Obviously, that's a terrible accident to have to deal with, but it's completely preventable if you pull your hair back and out of your workspace. I'll share with you, there was an accident at UCLA a number of years ago. Um, and I know about this because I have worked at Keene State as in the environmental health and safety department as sort of a temporary job for a while. I worked as the chemical hygiene officer. And a story was shared to us out of UCLA of a graduate student, of all things, who accidentally set herself on fire in the laboratory. And sadly, no one in the laboratory responded quickly. And this poor woman, the fire spread because not only was her hair flammable, but her clothing caught fire. And she was panicked. So she just stood there, and her colleagues didn't think to get her to the emergency shower, or they just froze. And this poor gal, she had such bad burns over her body, she eventually passed away from them. So it doesn't matter what kind of lab you're working in. It doesn't matter how experienced people are. you have to always be thinking about the dangers that are, or the hazards that are around you, and behaving in a way to keep yourself safe. Um, that's just a really important message. So I mentioned gloves a couple minutes ago. Gloves are the one piece of PPE that people use in all labs. Again, during the pandemic, people have had to learn a lot about PPE, right? People have had to learn about masks over this past year. Um, so things like lab coats, eye goggles, face protection for your eyes, face masks, these kinds of PPE are going to be used when they are needed. And there are certain laboratories that are doing work that requires eye protection or requires face masks and even beyond. It depends upon the hazards that are associated with the work. You'll see I have the term BSL here. We'll talk about this in just a minute. Laboratories that handle living material, so microbes, like a microbiology lab would handle, cells. Many labs grow cells in culture. Any kind of tissue. Um, all of those labs get rated based on their hazard level. And they have to use PPE appropriately to keep everybody safe. But gloves are always used, whether you're in a micro lab or a lab that handles, again, living materials, living cells, or you're in a chemistry lab or any kind of lab at all. Gloves are always being used because gloves help prevent all different kinds of exposures. But we have to be careful that we put them on appropriately, that we take them off safely if they come in contact with anything dangerous. Here are some of the things we need to remember in the micro lab. When you put your gloves on in the micro lab, once they're on, you have to think to yourself that you will not touch anything that is not routinely disinfected. Now, what do I mean by that? I mean, once your gloves are on, you're not going to want to touch anything that is not going to get disinfected later on. So, for example, you're not going to want to touch the microscope with your gloves on. You're not going to want to touch a computer keyboard with your gloves on. You're not going to want to touch the doorknobs to the lab with your gloves on. Okay. Those things are not, disinfected every single day. So we have to always be thinking that when our gloves are on, we're not going to touch those things. If we need to touch those things, we're going to take our gloves off. Remember, we don't reuse gloves. These are considered single-use items. If you take a pair of gloves off, you throw them away and you get a fresh pair. We also don't leave laboratories with our gloves on. I said a minute ago, you wouldn't want to touch the doorknob with your gloves on anyway, but you certainly wouldn't go out into a hallway with your gloves on because, again, your gloves might be contaminated with something. So you wouldn't wear your gloves to go to the restroom or to go down to somebody's office. You would take your gloves off. It's really important, as I said a moment ago, to know where all the safety equipment is in any lab you're working in, the eye wash station, in case somebody splashes something into their eyes, the emergency shower, as I said, in case someone accidentally gets set on fire, even the first aid kit. You should know where the first aid kit is in the lab, just in case somebody cuts themselves. Know where the fire extinguisher is in the lab. The saddest stories are when something happens in a lab, some kind of an emergency happens in a lab, and nobody knows what to do because nobody knows where this stuff is. So it's kind of on us as the people who work in labs to familiarize ourselves with where all the emergency equipment is, just in case. And that way, if an emergency happens, we can know exactly where to go to get what we need. So when you put those gloves on in the micro lab, or any lab really, you're going to treat them as though they're contaminated with something, even if you know they're not. Even if you know you come in, you disinfect your work surface, you put your gloves on, you know those gloves aren't contaminated yet, but you're going to act as though they are, because that way you will always act as though they're contaminated. You don't want to touch anything that is not going to be routinely disinfected. So you don't want to touch your face with your gloves on. If you're like me and you wear eyeglasses, don't touch your eyeglasses. Don't touch your hair with your gloves on. Certainly don't touch your personal cell phone or your personal laptop or anything like that. You don't touch microscopes. You don't touch textbooks or door handles. Anything like that that's not going to get disinfected every day, multiple times a day. You just take your gloves off. If you need to scratch your nose, take your gloves off or take at least one glove off and scratch your nose and then get a fresh glove and put it on. Um, Lots of people in labs, including in diagnostic labs in hospitals, use something called clean hand, dirty hand technique. Um, if you think about it, sometimes we need to do things like type while we're doing our work. Maybe we're recording data while we're working. If that's the case, you keep one hand gloved. That's going to be your dirty hand. And you take the glove off the other hand. That's going to be your clean hand. So let's say I was working and I was doing some kind of experimental work or diagnostic work. I would do the actual procedure with my gloved hand, my dirty hand, and I would use my clean hand to write down something or to type something into a computer. And finally, when we're done at the end of the day with our gloves, or we change our gloves during the workday, gloves are considered non-contaminated waste. Now, I know I just said to you, I want you to treat them as though they are contaminated. But even if microbes get on a pair of gloves, even if we were to accidentally get culture onto these gloves, the thing is, the cells won't survive. Between the material that the glove is made out of and the fact that there's no water, there's no nutrients for the microbes to eat, any cell that lands on this glove is going to die pretty quickly. So at the end of the day, we throw them in the regular trash. Here's a couple of really important things to remember, and you'll see this discussed in this week's lecture topic as well. Microbes are all around us every single day of our lives. Our body is covered in microbes. Our external surfaces are covered, our internal surfaces are covered in microbes. The environment around us is covered in microbes. The thing to remember is that most microbes are not harmful to us. And of course, some of them are actually helpful. They're actually necessary for us to stay healthy, right? We're covered in some microbes that are necessary for our health. When we work in a micro lab, we are working with cultures full of microbes. They're everywhere. Even if you just work in, say, a healthcare setting and you're not handling microbe cultures, but you're handling patient materials, you're handling body fluids, you're handling urine samples or blood collection tubes or biopsy samples, you have to assume that those things have the potential, at least, to be contaminated with microbes. They are everywhere. Luckily for you and me, most of them are not disease causing. Most of them are not pathogens, but there are pathogens out there. So we have to always protect ourselves. We have to behave as though everything we're handling is dangerous to us. every tissue sample, every urine cup, every blood tube. We have to handle it as though it contains something that can harm us, some kind of pathogen. Down at the bottom of the slide, I want you to put a star next to this in your notes. The two most important things to remember. So, the most important things to remember about working in a lab that handles microbes: don't contaminate the culture, the microbe culture, or the diagnostic sample with yourself. You're covered in microbes. So don't get your microbes into that pure culture or that patient's diagnostic sample. And number two, don't contaminate yourself with the culture or the diagnostic sample. Don't contaminate the culture with yourself, and don't contaminate yourself with the culture. If you can keep those two things in mind while you're working around microbes or potential microbes, you'll be in really good shape. Anybody have any questions? You know, it sounds like a lot of sort of rules and regulations, and it might feel like, oh, I can't possibly learn all of this, or it would be really hard to remember all of this at work. But I want to assure you that once you begin working, and you start to get accustomed to your job responsibilities, whether you're working in a lab or whether you're just coming in and out of a lab periodically during your day, you get used to these things very quickly. First of all, the people in the lab are hopefully doing all of these things, and they will let you know if you're doing something incorrectly. Um, and the learning how to think in a way of safety, of biosafety, becomes a habit after a while. Um, for example, I want you, when you leave this course, I want you to feel like you would never pick up a culture without gloves on. Like that would feel so abnormal to you. That's how I want you to start to feel at the end of this semester. You would never pick up a petri dish with your bare hands that had bacteria growing in it, or you would never pick up a urine sample without gloves on. Um, I want to keep reinforcing these biosafety things for us so that they become second nature. That's really the trick to it. You want them to become second nature to you so they feel very natural. All right, I'm going to dive back in here. I mentioned a couple of moments ago that any laboratory that's handling microbes or handling cells or handling human tissue, basically, or animal tissue for that matter, any kind of living material, it's going to get graded according to a biosafety level. This is called BSL, and you may have heard of this before. You may have worked in laboratories that had their BSL level posted for you to see. All this is a system of standards that are applied in order to keep the lab personnel safe and the public safe. So these biosafety designations, again, or biosafety levels, as we call them, are abbreviated BSL, and they range from number 1 to number 4. So number 1 is the lowest level of hazard, and number 4 is the highest level of hazard, just like that door sign we talked about earlier. Now, there's a lot of information on this slide about what is required in these different BSL level labs. You are not responsible to learn this. Okay, I am not going to ask you, the specific regulations for a BSL-3 lab. Here's what I want you to know. I want you to know that a BSL-1 lab has the lowest level of hazard associated with it, and a BSL-4 has the highest. I also want you to know that as you go up from one level to the next, all of the regulations from the previous level still apply. And then there's going to be some new regulations. So, for example, if you're working in a BSL-2 lab, you're expected to follow all of the regulations for BSL-1 plus some additional regulations. Now, I want to share some pictures with you because one of the biggest differences that you'll notice about these labs is what the PPE is that's appropriate for the lab. Um, I'll also mention that I should also mention to you that students sometimes get confused about the difference between a BSL-3 and a BSL-4. If you read through what the requirements are, they sound very similar. And again, I'm not going to test you on what the requirements are. But if you're curious, the biggest difference between a BSL-3 lab and a BSL-4 lab is whether or not, if there was an accident with a microbe, whether or not there's any treatment available or any vaccine available. So BSL-3 and BSL-4 labs handle the most dangerous pathogens. But the difference is a BSL-3 lab handles pathogens where there is either a treatment or a vaccine available. These are typically respiratory pathogens. So pathogens that affect the lungs, the respiratory tract. In a BSL-4 lab, they are handling the most dangerous pathogens, including, again, respiratory pathogens. So right now, for example, in our country and around the world, the virus that causes COVID-19 is being handled in BSL-4 labs. Now we do now have vaccines available, but until those are widely available, all of the research that's being done on the SARS-CoV-2 virus, the virus that causes COVID-19, all of the research that's being done is being done in BSL-4 labs because that virus is still considered very, very dangerous. But eventually, once there's plenty of vaccine and hopefully maybe some good treatments available, that virus will move down a category and people will be able to study it in BSL-3 labs. That's where influenza is studied, for example, in BSL-3 labs. There are many fewer BSL-4 labs in our country than there are BSL-3. I believe we have one or maybe two BSL-4 labs down in Massachusetts doing really important work. But as you might imagine, since they're handling such highly hazardous microbes, they're not very common. So take a look at these images. On the left-hand side of the slide here, you're looking at someone who's working in what would be a BSL-1 lab. All educational labs, all of the labs on college campuses, for example, are BSL-1. We do not handle pathogens in those labs. So if we were on campus right now and we were sitting in the classroom, it would be a BSL-1 space. You'll notice that she does have some PPE on, right? She's got some kind of it looks like scrubs that she's wearing. Maybe that's required by the lab where she works, but she has her gloves on. She's got her hair pulled back. She's doing all of those sort of general things that she needs to do. She's got her workplace organized, general safety standards. This woman is working in a BSL-2 laboratory. You'll notice this gigantic device right here. This is called a biology cabinet, or you'll hear it called a biosafety hood or a biosafety cabinet. This is not the same as a chemical fume hood. If you've been in a chemistry lab, you know that they have fume hoods. They have these compartments that are there to help draw toxic fumes away from the person who's working. But a biosafety hood or cabinet is designed to maintain a sterile surface. So there's a glass door that comes down. You can see her hands are just through the crack under the door. And she's working on a surface that is kept clean, it's kept sterile with the use of air currents in here and ultraviolet light. Now she's doing something very naughty in this picture. Can anybody pick it up? What has she not done that she
Not
should
put our
do?
hair back.
She does not have her hair back. I think they took this picture for educational purposes. So yeah, this is not typically a problem that you see in BSL2 labs. I think this picture was taken on purpose, but yeah, she didn't pull her hair back. Very naughty. All right. Now on this next slide, on the left, this person's working in a BSL3 and this person is working in a BSL4. So obviously, when you look at the BSL4, this is the most extreme PPE people can wear. This is a self-contained suit. It has its own air supply. So this red coiled tube is pumping air into this suit. So this person is not breathing room air. They're breathing the air that comes through this tube here, this red tube. Remember, these are the worst pathogens we know about at any moment, and there is no treatment for them. There's no vaccine. If there were an accident and this person were to get exposed, we wouldn't be able to help them. So that's why they wear these self-contained suits. She's also working under a biosafety hood. You can see the glass door has been pulled down over on top of her wrists. So all of the previous levels of safety are still being used in addition to this very special PPE. Now this person here is in BSL3. So she is breathing room air, but she has a mask on. You'll notice that all of her hair is not only up, but it's covered. She's got multiple layers of protective clothing on. She's got her gloves on. She's working in a biosafety cabinet. Again, the biggest difference is that BSL4, you have to wear this self-contained suit in order to work. You can imagine that if at BSL4 labs, very few people are actually allowed in to the lab space. You need a lot of training for those. I have been in a BSL3 lab, but I've never been in a BSL4 lab. Okay, so let's talk about some things that are specifically related to the micro lab. Always assume that work surfaces are contaminated while they are in use. Now that seems weird. I told you that you're going to disinfect your workspace before you begin working. So you know it's clean, at least in that moment, but you're going to work through the day as though it was contaminated. And what that means is you would never place anything uncontaminated down onto your workbench. If you're holding something sterile in your hand, you are not going to put it down on your workbench. If you did, you would have to assume that it was contaminated. Similarly, you're not going to put anything contaminated down on your workbench either. So for example, inoculating loops. These are a piece of equipment that we use all the time in the microbiology lab. We're going to take a look at those later on. The caps off of test tubes, the lids off of Petri dishes, syringes, needles, anything that we're handling that is contaminated or potentially contaminated, we're not going to put down on our desk. Because if we do, now we know it's contaminated, or now we're certain that it's contaminated. So we assume that the workplace is contaminated. We don't put anything down on the workplace that is uncontaminated or is contaminated, known contaminated. Okay. Whoops. One other thing on this slide to remember. This inoculating loop is essentially what allows us to transfer cells, microbe cells, from container to container in the lab. We have to decontaminate, we have to sterilize that loop over and over and over again during the day. We have to decontaminate it both before we use it so that it's sterile, and after we use it so that it is sterile once again. So we are constantly sterilizing these loops all through the day. And again, we're going to talk about that more, that process more in a later lab. We use heat to decontaminate these loops. Obviously, flames are common, Bunsen burner flames. You can stick the metal loop into the flame and heat it up to sterilize it. We also use devices called incinerators. We place the loop inside the incinerator to heat it up. In some labs, you'll even find what are called alcohol lamps. These are not Bunsen burners. They're not burning a gas. They're burning alcohol. And you get a flame out of the top of the lamp that you can use to sterilize your equipment. You don't see alcohol lamps very often anymore, but they are still around in some labs. What else do we think about when we're in a micro lab specifically? I said a few minutes ago, you would never pick up a culture container without gloves on. So if it's a test tube and you're growing microbes in the test tube, or if it's a Petri dish and you're growing microbes on that Petri dish, you always keep a gloved hand when you're handling culture material, just in case some of those microbes are on the outside of the container. Test tubes, just a small thing here, but test tubes are always kept in a rack. You would never lie a test tube down on a work surface because of the risk of spillage. And something else that might seem odd, but we always keep our Petri dishes upside down while we're working on them. And the reason is these dishes have a solid material in them that we grow microbes on. It's called agar. So we spread some microbe cells out on this solid material and we let them grow in the incubator. If you have this plate right side up, so the lid is on the top and the agar is in the bottom of the dish, what tends to happen is condensation tends to gather on the lid and it will drip down on top of the agar and it will interfere with the growth of these cells. So what we do is we just always keep these plates upside down. We keep the bottom of the plate up on the top and the lid down on the bottom. And that will prevent any condensation from interrupting our microbes while they're trying to grow. And lastly on this slide is this thing right here. This is an autoclave bag. These are the bags that we put things in before we put them into the autoclave. So remember why we use an autoclave. We want to sterilize something. We want to kill all potential microbes. In a hospital setting, we package up things like surgical instruments and we put them into the autoclave to sterilize them before surgery. We sterilize surgical gowns. Surgical gloves get sterilized. There's all kinds of things that we need to sterilize before we use them. IV catheters are sterilized. All kinds of materials in the hospital have to be sterile when we use them. In a micro lab, we're not so much sterilizing things so we can use them. Rather, we are sterilizing things because we know that they're contaminated with microbes. So we're trying to kill the microbes before we dispose of them. So these culture tubes, for example, we know there are microbes growing in here because we put the microbes in here. We know there are microbes growing on this plate because we put the microbes on the plate. So when we're finished with these disposable culture containers, We put them into this bag and we put the bag in the autoclave and we sterilize these containers. We kill all the microbes so that the bag can then be thrown away. Or I should say the things in the bag can be thrown away. This symbol right here that's on the front of this bag, this is a biohazard symbol. Anytime you see this symbol, it means that whatever's in there, whatever kind of container it is has a biologically related hazard to it. There's something alive or associated with living tissue that's in there and is hazardous to us. And the last slide for today, before I let you go, just has a couple of summarizing statements on it. Remember, all teaching labs are BSL-1 labs. So any teaching lab, again, that handles microbes or handles cells or handles tissue, any type of living material is, if it's a teaching lab, it's BSL-1. And the important thing to remember about a BSL-1 lab is that no pathogens are handled in that lab. It's also important for us to remember that most microbes, the vast majority of microbes in the world, are not pathogens. Only up to about 5% of bacteria and fungi and other types of microbes are pathogens. The rest of them are not able to cause disease. So that's a good thing. We're going to treat all microbes as though they are pathogens, though. We're going to handle every microbe as though it is able to cause disease in us, because that way we'll always be using our highest safety standards. Here's something that you'll hear me say a lot this semester. When it comes to microbes, microbes don't fly. They don't fly up. They fall down. Now, what do I mean by that? Why does that matter? Well, it means that if I open up a petri dish that has bacteria growing in it, the bacteria that are growing on that dish are not going to fly up and contaminate me. The only way for me to get contaminated would be for me to touch it. But something could fall down out of the air and land on that dish and contaminate that dish. Microbes fall. And in the micro lab, we have to be careful that we don't allow microbes that are in the air to contaminate our work. We have to protect our pure cultures from getting contaminated with other microbes, because remember, microbes are everywhere. So again, we'll be hitting on all of these things during the semester again, but today we just want to introduce them so you can begin thinking about the techniques that we use, not only in the microbiology lab, but in all labs to help keep us safe, number one, and keep the public safe, keep the environment safe, keep people outside the lab safe. All right, who's got questions? Any questions at all? Any questions about the biosafety material or about the course? Any questions about what you need to do this week? Remember, you can pull that to-do list up anytime you want in Canvas.
I just had a quick question. I got
Yeah.
an email about course materials, like it was through the first day program for this course, and I wasn't sure if that was something that we needed to get or not.
I'm wondering what that is. The first day program?
Yeah, it says Mastering Microbiology with Pearson eText, and it's like through the, I don't know.
I'll tell you, I suspect there is another section of this course being taught by Dr. Boyer, Janelle Boyer. Her class is a little different from mine. They're both microbiology, but we teach them a little bit differently. I suspect that came through, it was intended for her students, but it came to all of you. That's what I suspect, because I know she uses Mastering Microbiology.
Okay, so we can opt out of that then?
Yes.
Okay, perfect. Thank you.
Yep. I'll bet you that's what that is. Yeah, I don't love mastering. I don't know if you've had any experience with it in other classes, but I prefer to make it's basically quiz material, homework material, and I actually prefer to make my own materials. So we don't use it in my section. Any other questions? All right, I'm going to let you go. Don't forget, if you have any questions, you can send me a message through Canvas. Any questions about what we've talked about today or anything else about the course, don't hesitate to get in touch with me. I will see you again on Thursday at 4 o'clock, and we will continue talking about this week's lab material. All right. If there's nothing else, I'm going to let you go. Have a good evening.
Thank
Thank
you.
you.
All right. We'll see you
Thank
next time.
Thank you.
Okay.